So if we were going to ask an optimist about where biochemistry comes from, okay, you can tell where my background lies. What are we looking at here when we asked our optimist what happened? What is all that? Organic molecules. That's biochemistry. Biochemistry is how do we take those individual pieces, arrange them into larger structures, and then how do those larger structures communicate with themselves to do really cool, larger aspect things. Okay? So that's where biochemistry comes in, is how do those molecules interact and talk to each other to build larger things. Okay? Which means we then need to be able to know what those things are. Okay? So for instance, if I took a look at that center structure, I know that center one's super obvious, that one, what does that mean? This goes back to what is our chemistry understanding? What does that mean? Okay, so we heard a couple terms come out there. We heard cyclohexane. We heard benzene. Anything else? Aromatic. How about sigma? How about pi? How about resonance? Okay. How about carbon, running out of space? Hydrogen? That simple drawing implies a lot of information. Okay? And hopefully we're all at a stage now where we recognize that information and we can see it and we can interpret it relatively quickly. Whereas if we try to do this class before any chemistry background, we'd look at it and be like, I have no idea what the heck any of that is. Okay? Kind of makes sense? Cyclohexane. Okay? It's not going to be a cyclohexane. Okay, so I heard one quick change. The ane implies single bonds or sigma bonds. We have pi bonds, so it can't be an ane. Okay? So I heard a quick adjustment to cyclohexene. That now accounts for pi bonds, right? But how many pi bonds does it take account for? One. Is there only one? No, there's three. So we would technically need to go through and actually change this all the way to cyclohex triene. Okay. It's good. We should all try. Yeah. Okay. Next part. Are all the double bonds at the exact same location? No, they're located at different positions. So we need to add more information about where all the double bonds are located. We got, what is it, 1, 3, 5, cyclohex triene. Ugh. That's why we call it benzene. Okay? So we have this knowledge. Some of it is readily available, some of it isn't. Adjust from what you've got. Ask questions when you don't know. Okay? As we look at all of these, some of them are going to be more important to know information about than others. Any guesses?
Alcohol.
Okay, and alcohol. Why is alcohol important?
Reactivity.
What reactivity? Numbing our brains after we finish this class. No. So the alcohol does have some hydrogen bonding aspects out here, okay, and that could lead to important chemistry. Okay. When you think biochemistry, what do you think of? Let's try that.
Cells.
Cells. Anything else? Proteins. Okay, that's actually the answer I was fishing for. Not that your answer was wrong, but I was fishing for proteins. Okay, what's another name for a protein? Or, sorry, how do we make proteins? Amino acids. Amino is and an acid is so functional groups that will play an important role. Okay? We tend to imply a lot of this knowledge in the name and then ignore what it means structurally. The name gives us some of that structural feature. I would argue the amino and the acids are going to be probably the two largest or most important functional groups to nail down. Because They are what define proteins. Okay, so they're really, really important. The other big ones that show up would be these two. Not that the aldehydes aren't important, or sorry, I didn't say that. The alcohols aren't important because they are important and we will see them pop up fairly frequently. Right? Large characteristic of our carbohydrates. Okay. What about those two? I muffledly said one of them. Okay. We have an aldehyde and we have a ketone. Okay. How can we get an aldehyde or a ketone? Too vague. Give me more than that.
Oxidation or reduction.
Oxidation of?
Alcohol.
Bingo! There's our alcohols. Reduction of? Acid. Okay. So we're really looking at interconverting each of those pieces to generate different chemistries that can get us to larger or different structures. Kind of neat stuff. Okay. So, you need to be familiar with your functional groups. You should be able to look at them and say, that is that functional group, okay, relatively quickly. The big ones we just labeled. Okay. Some common themes that pop up. Okay. We get the same types of kind of biomolecules that you might see in organic chemistry, the functional groups. Okay. We can string those functional groups into slightly larger structures, and because they are now larger structures, we then kind of irritatingly change the name. Okay. So not only do we need to know the functional group of what an acid looks like, but when I combine that with an amine, I now have an amino acid. Okay. So it changes some of our characteristics. Okay. Everything that we're doing in biochemistry is about how we live, and how we live is about the consumption or production of energy. Okay. Which is ultimately tying back to physical chemistry. Okay? Probably none of you have taken what you would have called a physical chemistry class. Oh, you did? Oh, crap. Well, I might die and sputter on that day. Our physical chemistry, the level that we're looking at within biochem is typically the general chemistry level. Okay? So we aren't going into the hardcore details that we might expect in a physical chem class. Okay? And then the last one we could really look at is a beginning. How did all of this start? Which is really kind of the philosophical aspect to this. So we look at biochemistry as a class, and we're really not looking at biochemistry. We're really looking at chemistry and philosophy. That's what biochemistry is. It's the bridge between all of that. And depending on which part of this we want to focus, we may see different aspects. Okay? So we'll wax partly philosophical at the beginning here. We're moving from the inanimate to the animate. Okay? We start with something that has no life that now becomes alive. How do we do that? Okay? Well, we just took a bunch of functional groups, and all of a sudden they became, boom, there's a human. Not quite. Okay? What happened to get us there? Which begs the question even further, did I start with just those functional groups? Probably not. I started with other compounds. So how can I start from what is inherently not organic to become organic? That's a pretty big philosophical question. Right? And that was what Miller and Urier both looked at with one of their famous studies. They went back and said, okay, what are the likely molecules that existed at the origin of all life? Okay, and they came up with a list. This is a list of those potential molecules. We have ammonia, we have hydrogen sulfide, carbon monoxide, carbon dioxide, methane, nitrogen, hydrogen, and water. Okay? There's been lots of debate over whether or not those choices were correct or not. Okay? But to a certain extent, that's sort of neither here nor there. Okay? Are any of those alive? No. So we're starting with inanimate, non-life existing molecules, can't support anything. Are any of those part of the larger structures in biochemistry? Well, if we broke them apart, rearranged them, and put them together, yeah, but do I have ammonia in my cells, just contributing to life? Not really. Okay? It's a building block that gets me there. So we're starting with something that has inherently no characteristics of what we would define and associate with life. And we're going to take all those things, we're going to put them into one pot, and we're going to say, boogada boogada, do something. Where's my human? Okay? That's really what they did. Okay? They took all those chemicals, condensed them into one big closed apparatus. Okay? But remember, we said energy. Energy has to be a part of this. We have to get that energy from somewhere. Well, where do we get it? Okay? Well, there's thermal energy. Okay? We're looking at the formation of planets. Okay? Well, do planets have heat associated with them? Well, if I stepped into a volcano, I would probably say yes. Okay? When I'm looking at the initial formation of planets, I've got lots of molten metal floating around. So I've got plenty of thermal energy. Okay? I have potential light energy, okay, coming from a sun system. I've got dry particles in the atmosphere. Okay? Those particles could become charged with different thermal or light energies. Once I generate charge, now what do I have? There's a really simple place to find charge in this room. Where is it? The electrical outlet. As soon as I generate charge, what do I have? Electricity. Okay? So I've got all these different forms of energy. So what they did is took them all, put them in one spot, and then said, here's a bunch of energy. Now we're going to do your stuff. Okay? And they just let this experiment run. A couple issues come up with this. Okay? What if this system was open to the atmosphere? Is it just those chemicals anymore? No. We're potentially introducing outside life into it. We don't want to do that. So we have to close the system down and make sure no outside stuff can get in and contaminate it. Okay, what's the problem with closing it down? If it's all closed down, how do you find out what's happening inside? You just wait for a human to explode out of the glassware? Okay. We have to have some way to open it up to relieve some of that. So we have to build our glassware with proper valves and systems to make sure that when we test stuff out of this, we're not introducing life, okay, or signs of life. Okay? So they built this very careful apparatus, and they just let it run. And every so often, they would test it. Okay? They started this, ooh, shoot, I didn't put it up there, somewhere in the 1950s. Anybody know? I think it's 1950s. I should actually know a whole lot more about it because this experiment is still running. Do you know where it's still running? UC San Diego, where I got my degree. Well, yeah. But yeah, it's still running. They're still testing it. They're still pulling stuff out to see what's going on with it. Okay. And they haven't found life. But what they have found is hydrogen cyanide and formaldehyde and some small amino acids. Okay? What is special about cyanide and formaldehyde? Both of them are very reactive molecules that can react with other structures to build larger and larger structures, like amino acids. Okay? So while we haven't proven the existence of life, okay, really no scientist in the right mind said a human was going to jump out of the glassware. But what we were looking for is just some bits and pieces. Because there's an issue of time associated with this. Okay? You all remember when you first learned about a mole? Like, oh, that was an easy concept. I totally understood that. Be honest, really. You're like, oh yeah, mole, that's totally obvious. 6.022 times 10 to the 23rd. That's obvious. Okay? Why was that so hard to understand? It's massive. When we're looking at biochemistry, where we are right now is not a short time away from these things. It's a stupidly huge amount of time away. We can't comprehend that time gap. Okay? Which means when we then try to comprehend it, we're screwing the whole system up. Of course we're going to not understand how it works because we can't comprehend the very first step of it, the time. Okay? That doesn't invalidate everything. That just means we don't know. So seeing anything come out of this that is even remotely approximating what we have as building blocks is insanely cool. Okay? And that was our Miller-Urey project. One of the things they were trying to do was look at amino acids. We have since kind of gone through developments of what makes life, where did life start. And one of the new theories is that RNA is actually the front runner for the origin of life, not proteins. Okay? So that doesn't invalidate their experiment again, because maybe once we have those amino acids, those can generate RNA, and RNA is what triggered life. The amino acids just helped make them. Okay? So we're just looking at the bits and pieces and how these things can scaffold. After that, that's a bunch of philosophy stuff that I don't want to worry about. So we'll move to the hard, concrete stuff that we know exists. Okay? So when we move into the chemistry, what we're looking at with all the proteins and amino acids and DNA and RNA are polymers. We're taking a bunch of small units and we link those small units together to form larger structures. This is a polymer. How many of you took a polymer class? Why not? Isn't this all polymers? Why are we not taking a polymer class? Okay. Because polymers, for some reason, haven't cleared that gap in becoming a majorly, and of course this is going to be awesome to say while recording, relevant field. Okay. They've been kind of divided out into biochemistry, into materials chemistry, that we don't really see that crossover. But if you look at the clothes that you're wearing, polymers. You look at your sunglasses, probably a polymer. Okay. How are those polymers different from the biopolymers? Okay. It's the individual structures and pieces that made those individual molecules. When we look at the organic polymers that we're making in the lab, say make your glasses, how different are each of the individual subunits or individual monomers? Anybody know? They're identical. We go into the lab, we mix one chemical, maybe two, and we make a polymer. What is biology doing? 20, 30? And it is able to reproducibly generate polymers for very specific tasks. Okay? We take two, and we screw that up pretty thoroughly. Biology is running at 20, okay? And some minor deference to biology. That's pretty cool. Except that biologists don't recognize that. They just say, oh, I have this. It's going to do something cool. Okay? So our big polymers that we've got, we already talked about one, are proteins, also known as polypeptides. Why? Because some chemists tried to get in there and break up and actually make some nomenclature. Okay? So poly for our polymer, peptide. Unfortunately, they didn't fix this because peptide is an annoying way of referring to an amide bond. Why don't we call it a polyamide? Oh, that's chemistry. Serious. Okay? We've got polysaccharides, also known as sugars. Okay? We're taking individual sugar molecules and we link them to each other to make polysaccharides. Okay? Or carbohydrates, or complex carbohydrates. We can take nucleotides. We can combine different nucleotides and we make nucleic acids, DNA, RNA. So these are ultimately all just tiny pieces that are now lodged together and generate these even larger structures that we then study and manipulate and try to do our best to understand. Okay? As we scale this up to larger and larger systems, that's where we move into prokaryotes and eukaryotes and mitochondria and endoplasmic reticula and chloroplasts and chromophores. And all of that, I don't like. We're going to be perfectly honest about it. It has very good relevance. It's just not a super large part of this course. Your textbook does talk about it. It is sort of part of our curriculum, and in that we're supposed to mention it. Okay, we should see this. And we will look at differences of what happens in prokaryotes versus eukaryotes, because we get slightly different things happening. Why would we get different things happening? Are they different cell? Yeah. Okay. They have different characteristics. Those characteristics force different processes to happen within them. Okay. If we scale that up further, this is an even more less favorite picture. Okay. We can subdivide out into all the different categories in biology. Cool, that's awesome, that's neat, that's pretty, I don't care. Oh, thank God, back to something chemistry related. Okay. We can now also take into consideration the energetics of biochemistry. Okay? And what we've probably all remembered from biology, even all the way down to the high school level, is that we're looking at ATP, okay? Adenosine triphosphate. It is that molecule that stores all of the energy, or most of the energy, and is the primary transfer of energy from one site to another, and allows us to really exist as we know it. Okay? So we want to be able to look at what happens to ATP and what happens to its structure and its, changes. Okay, so we mentioned structure. How many carbons are in ATP? Because with the formula, right, we've got the information. No, there's no formula. Well, with the structure, no, there's not really a structure there either. Okay? We simplify down to the smallest piece that we can work with. If we looked at the structure or even the formula, it ends up getting too complex, so we skip it out to ATP is just the symbols. If we react that with water, what happens? We get rid of this big old piece. Thank you. We have a structure. And that structure we know as phosphate. Well, if I've removed a phosphate from adenosine triphosphate, I now have adenosine diphosphate. If we go through to balance, and of course we can balance a chemical reaction given this don't have the structures, H plus also comes out of this. So we now need to think about what things are going to contribute to this and how that contributes to our overall understanding of biochem. And with that, I will have to stop the video, and we'll pick up on Tuesday.